Get to IND faster, accelerated and high-performance cell line development at Samsung Biologics. Hello, everyone, and thank you for joining us on our webinar today, Getting to IND Faster, Accelerated and High-Performance Cell Line Development at Samsung Biologics. My name is John Gill. I'm the Director of Cell Line Development here at Samsung. Prior to Samsung, I spent 24 plus years in the biotech and pharma industry, with most of my time spent in cell line development, upstream process development, a little bit as a preclinical program lead, and certainly with expertise in the monoclonality and single-cell isolation field. All right, the contents of today's talk are in two main areas. One, the CMC activities needed for your IND in the fast timeline here at Samsung. And the second topic, how we use the cell line or accelerated and high-performance cell line development to getting you to that IND faster. All right, so next we'll move into the CMC activities needed for the fast IND timeline. Your success to IND, faster and better. The development pathline from gene to IND at Samsung Biologics starts with receipt of your gene of interest. The transfection, initiating the cell line development process, really kicks off that IND timeline. From there, we move to the MCB banking and characterization, integrated and simultaneous upstream, downstream formulation, and analytical method development. The non-GMP DS and DP production, which would initiate and facilitate the IND stability and taxot. Then moving forward through our capabilities is the cGMP DS manufacturing and GMP DS stability studies, both DS and DP, and culminating in the IND filing support that we offer here at Samsung. So the fast timeline, as we look at the critical path and what's required, this is a very customizable program or process here at Samsung. So reaching out to our business development colleagues at the beginning can lead from CLD to DP manufacturing in as little as 10.5 months. The initial contract discussions initiate with the gene of interest or your sequence and transferring that for the vector construction. The timeline truly kicks off as we initiate the cell line development process, which for most programs is a 3-month overall timeline. From there, we move into the cell line stability program, which is 2.5 months for 60 generations. Simultaneously, we transfer the research cell banks for the pretesting to initiate the master cell bank prep, as well as the upstream process development activities, initiating with clone feasibility in the 2-liter run, and ending with the tox 200-liter run. As we initiate or complete the 2-liter runs, the material generated there can initiate the DSP formulation and analytical program timelines. From there, the timeline initiates for the manufacturing settings. So the initial 1.5 to 3 month characterization studies required for the analytical method transfer, and the cGMP drug substance manufacturing at the 9 to 10 month time frame. This also moves to the 10.5 to 11 month for DP manufacturing and release, again with the regulatory documentation support starting prior to the DS manufacturing and culminating a little over a 12-month timeline. Moving from the required CMC activities for the timeline, we're going to transition into the accelerated and high-performance cell line development services that we offer here at Samsung. Accelerated cell line development through cutting-edge technology at Samsung Biologics is driven by the Berkeley Lights Beacon, where we have the optofluidic technology allowing us to fully automate the live cell isolation, selection, and screening in nanopens. We load over 5,000 individual cells, screening by specific growth rate and specific productivity, making the ranking and scale-up process much more efficient, where we're choosing the top clones right up front. How that integrates in the overall 3-month timeline for cell line development is initiated through the first transfection, selection in mini pools and stable bulk pools, initial screening in deep well plates where we can assess titer, growth rate, and critical quality attributes, loading those clones onto the beacon up to 6,800 individual clones, then pulling 24 to 48 clones into the deep well plates and shake flasks, allowing us to move quickly through that initial scale-up, moving to the Amber 15 for final clone ranking, where we freeze the top 5 clones for a research cell bank or your pre-master seed bank. This overall process prior to the beacon is about 6 weeks, which allows us to rank for a growth profile, productivity, and product quality. The timeline on the beacon itself is anywhere from 3 to 5 days, depending on the growth rate on the chip itself. And the final scale-up and screening is 7 weeks from the beacon to your research cell bank. The next part of the talk is to discuss the internally developed Samsung Biologics platform, the CHO K1 GS knockout cell line that was developed here over the last 2 years. The initial knockout of the glutamine synthetase gene was done with TALEN, and we went through 3 ranking stages or screening steps. 2 phenotypic screens where we're looking at growth with or without glutamine, and then that is sandwiched around the screening or the sequencing of the GS gene to confirm true knockout of the glutamine synthetase gene. The last stage of this is media adaptation. I'll go through this on the next slide. It's truly more of just a media change, as you will see as we go further. As I said in the previous slide, the media adaptation is truly just a passaging of the top clones here. So we took the top 16 or top 19 clones, passaged them every 3 days, seeding at 500,000 cells per mil. Over the course of 1 month. Here we're looking at the average doubling time and the peak VCD at that last fed batch stage starting at a little over a month. As you can see early on as the first couple of passages, we see no true drop in the doubling time or in the viability, really saying that this was more of a just media change than media adaptation, ultimately leading to the selection of the top 5 individual host cell lines that could be used for our performance test. The doubling time for these clones averaged between 18 and 21 hours per generation, and the peak VCD after a 6-day fed batch culture was anywhere between 6 and 8 million cells per mil. At this point, the media had changed from the CD-CHO to the XL CD-CHO fusion from Merck. Those top 5 clones, as I said earlier, were cryopreserved, and then we moved to the performance test for IgG1 expression. Once we had cryopreserved the top 5 potential host cell lines, we went through an individual performance test workflow. Here we're expressing an IgG1 and going through the entire cell line development platform as described earlier, where we did transfections and selection in mini pools, so 10 96-well plates. Those individual colonies or wells were screened by octet, where we carried the top 10% for a 24-hour QP assessment after the initial scale-up in suspension and deep well plates. The top clones the top mini pools from there are again scaled up for an 11-day fed batch in deep well plates, culminating in the top 5 being screened by a 14-day fed batch study. Again, and what I want to point out here, there was no vector development at this stage, so we're really just using a GS selection with the heavy and light chains driven by a mouse CMV. So no vector development had been done at this point, and we'll talk a little bit about some of the improvements we've made to that later on in the webinar. The first stage of the performance test ranking workflow is screening of the mini pools and the 96-well plates. And you can see right up front, initial clone 44 and 49, the transfection efficiency really gave us a red flag on 2 of the 5 clones, as this is not something that would be robust enough for us to offer to our clients as a host cell line. That being said, we still took the colonies that were generated and scaled them up through the 24-hour and 11-day and 14-day fed batch study for completeness of the data, but knowing that because of the viability or cell growth or selection efficiency, these were not clones or host cell lines that we could take further. The performance test, the primary screen, so selecting the mini pools that we want to put forward in the single-cell cloning, you can see very early on in our development workflow, we could see some promise of the hosts that we had isolated and generated. So the clone 48 and 56, yellow and green, you can see both the mean and the peak titer here, screened by the octet, mg per liter, is an improvement over our CHO control. We took the top 10% of these pools and moved them forward into the workflow screen. So jumping through our workflow or the performance test, the 14-day fed batch production study performed in shake flasks. We took the top 5 pools from each of the CHO control, the clone 48 and 56, and went through a 125 ml shake flask with a working volume of 50 ml through a 14-day fed batch study. You can see in the left part of the graph, the CHO control after the 14 days is a 2.9 gram per liter, which is typical for the IgG1s that we express here at Samsung. It's a decent expression after the stable pool stage. But comparing that to the 2 hosts that we had isolated and pulled through the ranking thus far, the clone 48 and 56, you can see a 24% improvement, so 3.6 gram per liter, and a 5.7 gram per liter, so almost 200% increase over the CHO control just at the stable pool stage. So when we talk about the accelerated timelines, having a titer of 5.7 at your stable pool stage allows you to accelerate your overall process development by initiating upstream process development, media screening, availing material for both downstream analytical and your formulation very early on this process. So on this next slide, we're going to talk a little bit more about the data from the 14-day fed batch process and really truly what makes the potential host that we're developing here quite special. You can see in the left portion of the graphs, the CHO control is a typical VCD and viability that you would expect over a 14-day fed batch process, with peak VCD being reached somewhere between day 7 and day 10. Afterwards, because of the drop in viability, you see a decrease in the overall viable cell density through the culmination of the 14-day fed batch study. That's improved quite significantly as we move to the right with clone 48 derived mini pools, where the viability does indeed drop for 2 of the 5 stable pools that were carried on, but there's a much better adherence to the peak VCD from that day 9 to day 14, leading to that 24% improvement in titer. But where you see the true improvement is in the pools that were derived from clone 56, where the viability, even at the end of the 14-day fed batch process in the stable pools, was in the 70s or greater. And that really meant that once you hit that peak VCD in that 20 to 30 million per mil stage, that VCD was maintained all the way through the 14-day fed batch process. There's many reasons why this could be important, both from an overall titer or yield from your process, but your product quality, because of that increased viability, will lead to better drug substance and hopefully. So moving through the workflow, we took the top expressing stable pools, as shown in the last 2 slides, and went through the single cell isolation. While our accelerated cell line development platform here currently is the Beacon, we do still use the single cell isolation platform of Cytina for isolation of the single cells, and the cell metric for imaging and confirmation of monoclonality for some programs if required, but there was certainly used during this performance testing. Here we took the top single cell clones and performed a 14-day fed batch as described earlier, this time with a change in the feed formulation. So this is Excel Advanced Fed Batch Media from Merck using a high clone feed, Cell Boost 7A and 7B with a 50 ml working volume. As described before, the CHO control itself in the stable pool, the maximum value was 2.9 gram per liter. The top titer from our CHO 56 host was 5.7. As we move to that single cell isolation and screening the 14-day fed batch process here, the CHO control yielded clones with a 3.4 gram per liter, so you still see an increase in expected titer from your stable pool to your single cell isolation, and that really held true for the clones that were derived from the CHO 56, where we isolated clones at 7.6 gram per liter. And again, I will highlight that this was done prior to any upstream or downstream or upstream process development or any work on the vector to this date. So now that we had clones that were reaching 7.6 gram per liter, we initiated some upstream process development. Here we're looking at just some feed strategies and different seeding densities. And we showed this slide really to point out the manufacturability or the robustness of the cell line, truly what makes the host special. You can see here where we did the study through a 20-day fed batch process, where the viabilities at the end of this study were still in the high 80s or 90% range, which is quite impressive. I will point out the viability drop around day 10 to day 11. That was primarily due to a change in the instrumentation used for cell counting. So we moved from one instrument to another, leading to that initial drop. But otherwise, you can see at the end of this study that 90% viability, which really allows us to hit that peak VCD somewhere between day 5 and day 9. And that is held all the way through a 20-day fed batch process. The end result here was 11.6 gram per liter. Now, while we show the 11.6 gram per liter and the improvement of the overall host cell line, this isn't a process we would normally recommend. We're just showing that at the 14-day fed batch process that is typical within the industry, you're not at the edge of failure. So you can expect run-to-run consistency and quite good drug substance product quality at the end of a 14-day fed batch process from run to run. So as I alluded to earlier, we hadn't done any vector optimization. So here I'm just going to briefly describe some of the preliminary work that we had done to improve the vector that was used for the performance test. So we took the PSBL1 base vector, which is a mouse CMV promoter, introns, the human CMV and human EF1 alpha. Cloned in here as a reporter for the vector screening GFP. We looked at a number of different promoter and intron combinations here. And as you can see on the right, we see quite a marked increase in the overall GFP expression. So 1.8 or twofold increase using GFP. For those of you that are familiar with vector design or vector optimization, you can do a lot of cheating with GFP. So do I expect that twofold increase would carry over from the data that we showed in the previous slide, that 11 gram per liter? Does this put us over 20 or 22 gram per liter? I will not even come close to promising that. But what it really feeds to is my confidence in this host and this vector design being a very robust system to be used for all of your process development and CMC activities. So what we'll talk about coming up is really then cloning out the GFP and looking at monoclonal antibody and Fc fusion molecules to really show true proof of concept with the change in promoter. So what did that look like once we cloned in the actual gene of interest? Again, I'm just repeating the data on the left here that was shown on the previous slide, where a twofold increase was shown using GFP from the PSBL1 to the PSBL9A. Once we use the IgG1 and Fc fusion, that same fold increase was shown with both molecules. So that gave us quite a bit of encouragement, both for our model using the GFP, but also that the improvements would bear fruit as we moved through now with an improved vector and improved host. The testing is continuing now, both with additional expression and stability enhancing elements, but as well as isolating these individual single cells from these stable pools, yielding the twofold increase to confirm product quality and stability in the cell lines that were isolated using this new vector technology. So to summarize getting to your IND faster, the cell line development process is over 2 parts. 6 weeks for the primary screen, where you're looking at generating the mini pools and ranking through deep well plates, looking at titer and product quality. The second stage over 7 weeks is the single cell isolation using the beacon from Berkeley Lights, the scale up to your manufacturability assessment and the Amber 15, leading to freezing or cryopreserving your top 5 RCBs in a 3-month time frame. This kicks off then the tech transfer for the master cell bank, and upstream process development. What this really exemplifies is excellent quality and outstanding capability. So as we showed here for the performance testing and some minimal process development both on the upstream and vector work, the monoclonal antibody titers of 11.5 gram per liter, additional molecules with bispecific titers over 4 gram per liter, and all of this with a faster and tailored timeline. This is supported by over 51 global regulatory approvals, with over 3,000 employees at Samsung Biologics, 1/3 of which are in the quality areas or quality functions, showing the commitment of Samsung to the quality really throughout the organization. It's a seamless one-stop service where it's fully integrated from cell line development through your DSDP manufacturing and IND filing. There's competitive pricing. As I mentioned earlier, it's a fast and tailored timeline to your needs. The pricing is both from a cell line development perspective, but overall through the process itself. So we guarantee competitive pricing with the best quality. And then maximum client satisfaction. We have dedicated program management. So each of the programs that we work with you on have both a senior and junior program manager to make sure that the communication is ongoing and constant, which really leads to the increase in your value from a faster timeline, higher quality, better pricing, and really that day-to-day or week-to-week communication, making sure that you're up to date and involved in the process that we drive here at Samsung. One quick note to point out is that once the current pandemic situation is complete, Samsung Biologics will be opening our first U.S. R&D center somewhere in the greater San Francisco area. I would anticipate this would happen sometime in the third quarter of this year. So as your most qualified, world-leading CDO, CMO, CRO partner, Samsung Biologics delivers your success to IND and BLA faster and better with maximum flexibility. We truly promise you the best. Okay, so let's move to the question and answer period. We have a number of questions. I'll say up front that if I don't get to all of your questions during this webinar, please reach out directly to our CDO business team at cdo.business@samsung.com or to myself at john.gill@samsung.com or hit me on LinkedIn as well. Additionally, if some of the questions that are submitted now aren't reached, we will reach out to you directly to try to answer your questions as quickly as possible. So let me go through the list here. What are the top important factors for this fast timeline? It's a harder question to tackle in this short period of time, but you can break it down into a few different points. You truly have to have a good plan up front with the QA, QC, and regulatory groups, both within your team and Samsung Biologics, to make sure that everybody's aligned with moving this quickly through your CMC activities. You have to have alignment both on the activities required and the milestones along that way. Really what makes that happen is a complete understanding of your MOA and your critical quality attributes, allowing you to design the specifications and process parameters so that you don't hit any hurdles or hiccups along the way. And then really what that means is working with a well-equipped CDMO that's flexible and that things will change through that process. So having good communication and capabilities with your CDO really would lead to success in driving those faster timelines. Next question. Important factors when choosing a cell line, like production, purity, etc. So I'll go back to the previous answer that I given. It really goes back to your critical quality attributes. You have to have focus on both your productivity and your product quality. And that's, I think, part of what is exciting about the internally developed cell line. So that having a host that's robust in consistency for run-to-run, high viability, that if you have a really good understanding of the mechanism of action and how that's driven by the CQAs, that's truly what's driving the cell line selection. Let's see, fast timeline applicable to other CHO hosts or other platforms. So if I understand the question correctly, we do offer support with two hosts here. So whether it's the SPL-derived CHO K1 GS knockout cell line or the chosen, the SAFC Merck host cell line, both would qualify, or we would work with both hosts within this time frame or these time parameters. So the platform itself is the same, both pre and post beacon, and using the beacon for the single cell isolation. So the timeline is applicable to both hosts here. This is a simple question. It's just pricing. Pricing, like I said, it's custom by each program. So I really don't want to speak too much out of term other than to direct you to our CDO business team to work out, based on your particular molecule, any of the analytics that's involved in helping us rank and screen both the stable pool and the single cells leading to your MCB candidate. That said, from a CLD perspective, there's no royalties or milestones. It's really just how you structure the overall development process here at Samsung on the pricing portion. Let's see. And next, any bispecific antibody experience. So what we showed here in the presentation was IgG1 and FC fusion data. We have additional development pathways with a bispecific antibody, but it's a little early in that from a product quality or even the titer standpoint to report. That said, we have extensive experience with, I think, over 25% of our programs at this point are bispecific, ranging from knob and hole and other more complex configurations. Typically, we see proper conformation anywhere from 3 to 4.5 gram per liter range with most of the bispecifics that we've been working on with our clients to date. So I think that's all I can cover right now. So as I said earlier, for the questions that have been already submitted, the CDO business team and myself will go through those. We'll respond as quickly as we can. If there's something else that comes to mind, please reach out to us directly, to the CDO business team or myself, either through the Samsung.com email or through LinkedIn. Thank you. Faster and better, your success to IND and BLA. For any questions, please contact the CDO business team or myself directly at cdobusiness@samsung.com or john.gill@samsung.com or through LinkedIn.